start. I know we're 4 minutes late, and in the name of time, we've got a lot to get through today. So let's go ahead and get started. Thank you for coming to the tech team. I know we had a crazy Great Salt Lake week last week at the Issues Forum, which was amazing. Thank you to Lynn. So this is kind of like the, after party, I guess, but maybe not as fun as some other after parties maybe you all had last week. But anyway, thank you for being here. I really appreciate it. I know you're probably sick of hearing about Great Salt Lake. Maybe not, because we all love Great Salt Lake. But anyway, let's go ahead and get started with some introductions. We'll just go around the room. I'm Marissa Weinberg. I'm with Forestry, Fire, and State Lands.
Ben Steierman, also with Forestry, Fire, and State Lands.
Tim Davis, Deputy Great Salt Lake Commissioner.
Mike Vandenberg, Utah Geological Survey.
Wayne Wurzbaugh, Utah State University.
Bonnie Baxter, Great Salt Lake Institute, Westminster.
Phil Brown, Great Salt Lake Brainstorm Cooperative.
Linda Freitas, Friends of Great Salt Lake.
Rebecca Downard, Utah Geological Survey.
Elliot Jagdecki, Utah Geological Survey. Andrew Rucky, Utah Geological Survey.
Jim Harris, One Poly.
Jamie Huff, Division of Emergency Management.
Maddie Gann, Forestry, Fire, and State Lands.
Let's go all the way to the back over there.
Leila Ahmad, Division of Water.
Jared Yanismaki, Forestry, Fire, and State Lands.
Connor Simon, DWR Great Salt Lake Ecosystem Program.
Scott Enoch, USGS.
Trey Trivedi, Bar Engineering. Casey Root, USGS.
Laura Vernon, Water Resources.
Marvin Hill, Fire Engineering.
Great. And then online, we've got Blake Bingham, Claire Spangenberg, John Luft, Kim Coburn, Kyle Stone, Tom DeMars, and Tyler McIntosh. Thanks again for coming out. So we'll just dive straight into it with updates from our various Great Salt Lake bodies. The Great Salt Lake Advisory Council, it feels like forever since we've met, and that's because we skipped our May meeting in lieu of the Issues Forum. So our next Advisory Council meeting is on July 10th, and it's at the Eccles Wildlife Center in Farmington. Really, our project load right now is small as we're coming to the end of the fiscal year. So we're still working on a water conservation toolbox with SWCA and the Division of Water Resources. And then we will hope to fund the mineral extraction study when you guys are ready for that. So those are the two final projects on the dock for the Advisory Council. And then we'll go on to the Salinity Advisory Committee, and our salinity guru, Ben, will.
That's a generous title. A very generous title. All right. So we have not met in a little while with the Salinity Committee because spring runoff and where we were at with salinity left us in a pretty good place. So the last volume weighted average was 115 grams per liter. And we had measurements in Gilbert Bay at around 92 grams per liter. So salinity's in a pretty good spot with this good spring runoff. I think we're going to continue to see it drop a little bit. And hopefully by the time runoff happens, we're still in a really good spot. But I think that our plan right now is not to modify the berm. We don't have any plans to go in and raise it or lower it. But we do want to see what's happened with runoff as far as any scouring or other structural modifications that may have occurred. So we've been working with USGS, with Christine and her group to do a survey of the berm once runoff is over, just to get an idea of where it's at. And then I think at that point too, we'll be convening as a salinity committee next month to see where salinity's at and try to make some future plans on, what, if anything, might need to happen this year. So I will also just mention too, as far as salinity and the north arm goes, the south arm is there's a pretty good head difference between the north and south, and that also means that the west breach, the lakeside breach, is fully communicating. Last I looked, I think it was around like 500 CFS, which is a pretty considerable flow. And we're also working with USGS to figure out how to maybe modify some of the channels that go out there to reduce sedimentation onto those gauges. So we're going to be looking at that. And then this wasn't on my update for the salinity committee, but I did want to say we are just starting our management plan process. I expect that the tech team will be heavily engaged, but for those of you that don't regularly attend, we'll be sending things out through our various channels through the tech team and the advisory council, and the salinity committee, So if you're not signed up for email lists, please get with Marissa and try to get your name there so we can make sure you get the information and how to participate. But that was all. Thank you.
Cool, thank you, Ben. So I'm sure what you've all been waiting for, some updates from tech team grants. So I'll start with fiscal year 24 housekeeping. Those that received grants for fiscal year 24 should have gotten a pestering email from me yesterday about getting your invoices in on time. We want to make sure you all get paid for the money that you were awarded. So please get those in before June 30th. Also, your final reports are due on or before June 30th. We understand that there's, different calendar issues with publishing and research and whatnot, but if you don't have a final report on that day, at least some sort of interim preliminary report, so we know that you're working on it and exactly what you're doing. And then a lot of you did respond to my request for filling presentations slots last time, so thank you for that. We're actually all booked up for 2024, which is exciting. I will start poking you all about 2025 soon. As it's still May, I don't really want to think about 2025 yet. So that's coming. And for our fiscal year 2025 grants that we just awarded, as you all know, we've received 22 proposals that totaled over $1.5 million, which is insane in a good way. I mean, it's great that so many people submitted proposals and want to do research on the lake. We were very excited and overwhelmed in a good way. So it was a really difficult ranking and awarding process, and the subcommittee are members of the tech team that gathered to read and rank and discuss these proposals. And we managed to get it all done in 2 hours, which was pretty cool. But with only $400,000 to award, it was very difficult. And so our division had to focus on projects that would directly inform our management of Great Salt Lake. Obviously, we'd love to give money to all the important research that goes on, but this time we really had to look at those priorities and focus our money efforts there. And so I'll just, before I let Ben talk about it, I was just going to read off the proposals that we did choose to award. Out of 22, we awarded 7. So as you can imagine, that was really difficult to kind of whittle them down to 7. So the first proposal we awarded was to Sageland Collaborative to continue their efforts with the Intermountain West Shorebird Survey. They're continuing a very important effort. They've been working on developing these surveys now for a couple years. We also just learned at the Issues Forum all the important work that they're doing on those surveys. So this grant will help them to continue doing the science to help us better understand critical shorebird habitat on Great Salt Lake. Secondly, we awarded 2 proposals to Sam Dutta up at Utah State. The first is to continue building his adaptive physics-based model for measuring flow through the causeway breach. So this final iteration of his model will really help us understand exactly how water is flowing through that tiny opening in the causeway. So we're excited about that. His second proposal is to build a 3-dimensional hydrodynamic model for quantifying heterogeneous mixing processes in the South Arm. So that's important as we are looking at where fresh water is coming into the lake and how it's mixing and how we can best control salinity. The next proposal was to Molly Blakowski from USGS. She'll be building a decision support tool to estimate PM10 dust emissions from the exposed lake bed of Great Salt Lake. So as dust concerns are rising in our community, and as our dust forum is happening this Thursday, Molly's decision support tool will be invaluable in helping us understand where dust is and how we can best manage it. Next, to the Division of Water Quality for investigating catastrophic die-off of submerged aquatic vegetation in Great Salt Lake wetlands. SAV is a critical food source for a lot of our wetland bird species, super important in the ecosystem. And so assessing the health of those plant species will be critical in maintaining the health of our wetland ecosystems on Great Salt Lake.
Thank you.
Next, USGS was awarded a proposal for more Great Salt Lake lithium resource investigations as well as groundwater inflow. So as we are getting more pressure and requests to extract lithium from Great Salt Lake, it's important that we understand where this resource is coming from, how it's recharging, how we're going to best manage our minerals. So another important proposal that'll hopefully build off of the good work Scott has already done on lithium. And then lastly, the seventh proposal was awarded to the University of Utah for mapping the lateral variability of groundwater input to Great Salt Lake using electrical methods. So this will complement the work that the University of Utah and UGS are doing with their current fiscal year 24 grant to investigate groundwater and how it's affecting Great Salt Lake water levels. So all of those 7 proposals, a mouthful, will help us to better our management as a division on Great Salt Lake. Certain things we talked about in ranking those in future iterations of this grant cycle, we understand it's important for proposal submitters to get some feedback from the committee. That's something that we want to build into this as we move forward in new years, to be able to provide that feedback so that those submitting proposals understand sort of where the committee went with their thought process and how they might reframe their proposals in future years. And then I'll kind of pass the mic off to Ben, who kind of, can describe a little bit more about our vision with this, this grant program and sort of how we're thinking of structuring it in the future.
Thanks, Marissa.
Yeah.
So I think the quick and easy version of this is that as you can see from the grants that we awarded, I'd say all of them are going to help us and our partners that are, have different management responsibilities on the lake, better manage the lake. And that was the goal. We really value all of the research that both this body and all of our researchers have contributed over the past, but there's obviously some priorities in the research now and the things that will allow us to make decisions better and more efficient. So we really want to move towards that in the future as well, and perhaps even hone in further on what those deliverables will be. So last year we redid the scope of, or the RFP, just, really hone in on what that management information will, or what that information will be that will better help guide our management. And we broadened the different areas. So we weren't as specific about what the hot topics were, to kind of see, what those areas of interest would be, and then really give that more weight to that actual management. But next year, I think that what we agreed to when we were ranking grants this year is that we're going to try to develop a little bit of a different process. I think it might look a little bit more like a procurement than a grant, just because I think we need to be there were a few proposals that we reviewed that had components that maybe veered outside of the specific deliverables that I think we needed. One specifically I can note to is just, we really want to do some brine fly, studies and understand that better. But most of the proposals we had some different components to the studies, and made it difficult to rank those and prioritize those against some of the other projects. So anyway, I won't continue to ramble, but I think if you're interested in participating on how we award grants in the future or award funding in the future, again, you can reach out to Marissa, but we'll be trying to convene that really probably this summer, so we're better prepared for the funding to be released by this time next year. Sure.
Could either of you comment on, there were some proposals, one, I think, Clark and Kettering, but maybe there were others that came to us, but we found funding elsewhere.
Sure.
Could you comment on those?
Yeah, I mean, so with limited funding, we're always probably hopefully we continue to get more proposals than we have funding. I think that's always better to have a lot of proposals and be able to handpick the ones that have the most impact. But there were some that we really wanted to fund and just didn't have the money. So like Karn Kettering's research is pretty integral to what, how our division manages Phragmites, and how the Division of Wildlife also manages Phragmites, and how we establish vegetation to compete with the Phragmites. And for a whole host of reasons, we wanted to fund that. So Keith Ambrick, our invasive species coordinator, is going to use some of his funding to help support that. There were some other funds in there that I know the commissioner's office had some interest in, and the advisory council has historically picked up some of those things. So when we get proposals that we can't fund, we typically still have a few that we would love to see funded, and we try to shop those around a little bit and maybe pique others' interest. So if you submitted a proposal and you weren't funded, all hope is not lost. I think we can still try to connect you with folks that might have some funding to support those studies. So yeah, anyway, appreciate that question.
Thanks, Ben. Yeah, if you ever have any questions on the tech team grants or what we're working on, or if you want to be a part of that, definitely reach out to me. I'm more than happy to throw you on a meeting or 5. So with that, let's get into our presentations, just so we give these presenters enough time to present. So Bonnie, if you're ready for us, I will share my screen.
Yes, ma'am.
So, we had a grant, way back during COVID from Forestry Fire State Lands, I think fiscal year 21. And we never really got a chance to talk it all through. And we've only recently processed some of the data in a big way, getting ready for paper. So I wanted to bring forth a few things from that one, as well as, let me make sure this goes forward. Oh, yeah. Hold on. Yeah, okay. I wanted to bring a few things forward from that past project, as well as the one that we had in fiscal year 23, which sort of extended some of that work and brought it into the lab for a little more isolation and studying these microbes in the lab. So, oops, I should just mention, Eric, I can't use my laser here, but Eric and Libby were in Eric Boyd's lab at Montana State University, and Eric Boyd and I have done a lot of work together on the microbialites over the years. I've worked with his grad students, he's worked with my undergrads, and we've had a long-term collaboration. Eric Dunham and Libby Phones have now moved to California. They have jobs and they've moved on, but I have their affiliation as Montana State because that's where they were when we were doing this work. Okay, so 2 things we were looking at. Really, the grant was about primary productivity, but as a control, we also looked at chemosynthesis. So most of us think about that old trophic diagram where, photosynthesis is the underlying bringing energy from the sun to all of us, whether you're talking about an aquatic system or a terrestrial system. But chemosynthesis in extreme environments is actually really, really important. We know that there's hydrogen metabolism in Great Salt Lake, for example. In these microbialite mats, there are bacteria, heterotrophic bacteria, and archaea that are probably just as important in creating food for the food chain. So we have this idea that brine shrimp eat Denaliella, and that's the food chain, and it's actually way more complicated than that. So I just wanted to throw this up there that chemotropy is something that we used also as a measure, and I'll explain that in a minute. But mostly we're focused on photosynthesis when we think about primary production, and we call the other secondary production. So both of these things are important in Great Salt Lake, and we don't talk about them enough, I think. So back in grade school, we all learned that photosynthesis is this chemical equation, and I actually worked with some teachers one summer, and one of the things that comes up on AP exams, the #1 missed topical area in biology AP exams in the United States is understanding of photosynthesis. So apparently we didn't get it. And so I thought maybe it would be instructive to explain what it is again, because you can say you got it, but maybe we didn't. So, whoops, I moved forward, I didn't mean to. So with photosynthesis, I'm apparently clicking when I don't need to, I'm sorry. With photosynthesis, those photons from the sun are creating sugar molecules. That's what's on the right side of the arrow, and then also oxygen. And so the sugar molecules, of course, get incorporated into biology, and that's what we think of as food for the system, and that helps move that food up the food chain. But also there is energy metabolism from the water that comes off and from oxygen. Oxygen's a really important electron acceptor, really, really important in the chemistry of cell biology. So if in the simple trophic food chain model that maybe we show the public, this really doesn't show up this complexity. So I wanted to say, let's forget about the shrimp and the flies and the birds for now, sorry. And let's just try to focus on the algae in the water column. There also are cyanobacteria in the water column. There are also algae in the mats. It's not just cyanos, it's very complicated. There's bacteria, there's archaea in both of those compartments. So this is a very simplistic view. And I want to, I've said this before, maybe in this room, but I want to underscore when people used to call cyanobacteria blue-green algae, and then we've got these other things that are more complicated eukaryotic cells that we call algae. That's why algae is actually not a taxonomic term. It has no taxonomy associated with it. And it's kind of a bad biology word, but we still use it in terms of things that do photosynthesis that aren't plants. Okay, so in this diagram, when I'm talking about algae, I'm really talking about microalgae, and inside them is something called the chloroplast, which is also inside of plants. Where did that chloroplast come from? Anybody know? Wayne knows. It used to be a cyanobacteria, and it crawled inside a bigger cell. So cyanobacteria are essentially chloroplasts, free-ranging chloroplasts. And they crawled inside a bigger cell at some point during evolution and gave that bigger cell the ability to do photosynthesis. So sometimes when we're looking at the genes of algae, we'll come up with the genes of cyanobacteria in the mix, and it's actually the chloroplast that's inside with its own DNA. So kind of wild, huh? Yeah, not that there's any evidence for evolution or anything. Just I digress. So these microbial mats, they used to be thick and dark and just really rich. And this is, I really appreciate Mike Vandenberg's wrist always in this picture that I show. Very nice wrist. That should be a wrist model, actually. But you can see just how thick and luscious these things were. This is when Mike and I really started studying these things like full throttle and bringing Eric's lab down and started having these big microbial convenings at the lake. But you also can see lots of brine fly larvae and pupa in this picture, which is pretty cool. So really a lot of biology in these things. And it turns out a paper we published in 2017 with Eric's lab, it turns out that a large percentage, forget what all the genera are that are on this bar, you don't care, right? But that's Euhalodece, the orange one, and we have isolated some of those in the lab. And if we ask what kind of bacteria are present, there's a certain gene that we use for barcoding that. And so this is Euhalodece, which now with the second phase of Forestry Fire State Land Study, we were able to isolate. This one we've not tried to isolate, but this diatom, this navicula, you might see navigation in this Greek root because it looks like a boat, right? So navicula are these boat-shaped diatoms, and they are also another important photosynthesizer. So back in 2017, when the lake was healthier, microbial mats were healthier, we saw these two major photosynthesizers in the mats, okay? We also did a project that was a result of Mike and I had a bet that all these different shaped microbes all around the lake were different. They were mushrooms, or they were ridges, or they were big domes. They were different, I thought, because there were different microorganisms present. And Mike said, no, it's the wind action. It's where they're located. It's the physical nature of the site. And I lost, and I had to buy him beer. But we published a paper together. And you can see in the biology, again, you don't need to know what these orders are of microbes. We can see the different colors. Every site we went to had identical microbial communities. So I totally lost. And the green boys there, that's the Euhalophyces. So that's that cyano that we found out was really important in the mats. So we did some work to find out who was there some time ago. And then in this grant that we had from Forestry Fair State Lands, initially, we wanted to understand who's doing more photosynthesis, the mats or the water column. And you can see in this picture, when the mats were healthy, you can see the green of the water column's full of photosynthetic microbes, right? And so are the mats. So who's doing it better, right? And so we did an experiment on the north end of Antelope Island, where we compared a site that had microbialites. We call that North Arm 1 up there. And it's actually Buffalo Bay, I think it's called, that is looking onto Fremont Island. And we also went to White Rock Bay. This part of White Rock Bay is kind of devoid of microbialites. Over here around White Rock, there's a big, nice reef there, but we sampled where they weren't. So if you look at my Solocator pictures that my students took, you can see that one area that we were sampling has a lot of microbialite action. So we waited up there and got some healthy microbialite mats. And the other one is this beautiful sandy beach. If you've ever been kayaking or paddleboarding out at White Rock Bay, it's just so fun. The sand is so beautiful. It's always ridgy like that. So just looking at, we're going to sample the microbialite mats here, and then we're going to sample the water here and the water here. Water without mats, water above the mats. That's the point. And what we saw in the mats is this community, Euhalophyces, pretty big, looking over 4 seasons. But check out the water, the planktonic communities, different species, different colors, right? Planktonic,
What's the y-axis?
that's just, that's an abundance measure. So how many pieces of DNA were present for each of those orders in this case? Yeah, we had to bend by order because there were so many. There's so many microbes in the water, I can't tell you. So we couldn't even go to genus. So that's the mats. This is right above the mats, the water column, different species. And this is right above, this is the water at the site without the mats. So the planktonic communities seem the same, whether they were above the mats or not. And when we did some work in 2017, we did find a lot of the microbes from the mats when we sampled the water right around. So I think there can be some sloughing off depending on the conditions, right? But we didn't see that in this study. So now we want to measure photosynthesis. And there is a proxy way to do that. That is sort of the historic way to do it, which is to measure chlorophyll. And there's a general relationship between how much chlorophyll is present and how much photosynthesis goes on. But the accurate way to do it is to measure carbon assimilation. But we can't throw radioactive C14 into Great Salt Lake. So we did this in microcosms, where we would take the water sample and shoot it into a vial, and then stop it by heating it. We boiled it to stop the reactions. So that was kind of the way we did this. So we're actually looking for incorporation of radio-labeled carbon into molecules, okay? But all within a microcosm. So the spring sampling, like the controls didn't work, so we kicked that out, and we've got 3 seasons. And what's interesting is the mats always show higher, whether we're talking about chemosynthesis, these are the ones we hid from the sun, okay? We hid these from the sun, we kept these in the sun. So chemosynthesis, mats are higher than either of the planktonic samples. And that was true over those seasons. And also mats are higher in photosynthesis than the water column in these experiments. So I'm using this to point to how important these mats are, right? And also notice in this one that chemosynthesis is probably as important as photosynthesis, right? So I'm going to go back to this other one. I forgot to say this. We don't see this incorporation in the winter. That's a really important point because the chlorophyll is very high in the winter, right, Phil? Chlorophyll is very high in the winter. We don't have grazers grazing the algae, right? But biochemistry really likes warmer temperatures. So if you look at photosynthesis articles where people have studied the temperature related for the enzymes to work, you actually see that photosynthesis is not happening at this cold temperatures. So that's one reason why chlorophyll as a proxy is a problem, because you can have high chlorophyll and no photosynthesis. Okay, so I wanted to point that out. In the summer, it probably relates, but in the winter, it does not. Okay,
One-
let me go. Sorry.
Bonnie, could I,
Yeah.
ask a question or make a point that the units in that one are per milliliter. Amount of photosynthesis per milliliter.
Yeah.
So,
Femtomoles,
pardon me?
femtomoles per milliliter.
Yeah, but how are you comparing a milliliter of algal mat with a milliliter of water?
Yeah.
For the water column? It's kind of an apples and oranges.
I'll send you the methods of the paper, but we actually.
Well, I know, okay.
I'll send you the methods because you're right, it is complicated. So we had to figure out a way to do that. And so we did dry weight of mats when we were doing, and we did, I've got to remember this, we did a normalization with chlorophyll. We did a normalization with DNA abundance. We did dry weight. There's a whole process we went through to equate those. So it's a really, really important point because you can't take volume of something and mass of something and make them, it is apples and oranges. So I'll send you the methods because I'd love your feedback on that before we submit the paper. Yeah. Okay, so the take home message really is when you extrapolate throughout all the seasons through the whole volume of the South Arm, and again, I'm only talking about the South Arm, of course, that the mats are contributing more, both in chemosynthesis and in photosynthesis, to the primary and secondary productions of the lake. And I think that's the important message here. And Libby, one of the former grad students, now postdocs, she got this calculation that it's, if you look at the whole lake, what the mats are doing for the whole lake over an annual period, that it's like the biomass of 7 elephants, which I think was amazing. This is why we don't want the mats to dry up. This is why we don't want them to, sorry, I went in the other direction. We don't want them to salt out, right? We don't want them to dry up. This is really important to keep water over the microbialites. So if you need data to support that argument in any venue, I'd be happy to share this with you. So we've pseudo isolated this Euhalothes species. We've been having some technical problems because it's very bound up with some bacteria and archaea that like to live. These guys live in communities. You know, they don't like to be isolated. They probably share metabolites. So we're having trouble getting the bacteria and archaea away. Every time we sequence the genes, we have other genes present. So, yikes. So, somebody was asking for an earthquake, maybe we'll get one. So those are the technical challenges that we're talking about. Once we get it clean, we really want to sequence the genome because that will allow us to search for genes that are involved in dehydration, rehydration kinds of pathways and understanding its resiliency and its salt tolerance. So to get the sequence of the genome is really important for understanding that in the lab. So that's why we're trying to isolate it. And the other one, oh, I should say, in the meantime, my students and I published a paper with Carrie Franz at Weber and her students on rehydration of the methods. And Carrie did this graphic, and I just think it's wonderful. It actually made it into our paper. But the idea is, hey, this is the healthy place. And when we start to lose a little bit of this was going on in 2022 when we were publishing this paper and doing this work. This is the north arm, right? We don't want to go here. We've done this experiment already. We know what it looks like, right? We want to shift back in this direction. So can these mats be rehydrated? And the work Carrie and I did in this paper shows that that's possible. The microbial communities can regrow, but they look different. And we're doing some current experiments in our lab that stems from this last Forestry Fire State Lands grant to show how that happens. Once we get here, all of those microbial communities are gone. When we sequence the microbialites in this area, there's no microbes there that are capable of doing the action of the mats for the lake. So these hotels are vacated. There are some people, still renting these rooms, but these are really full and healthy. So we'd like to go this way, right? One of the things that I want to end with is this cool microbe that we have isolated from the lake. So as we took dry microbialites, they were dry on the beach in 2022. They sat in the lab for 6 months. And then we're like, I wonder if we could rehydrate those guys. I wonder about what happened. We put them in Great Salt Lake water from Antelope Island. And sure enough, the mats started to regrow. And then we sampled the mats and we asked the question, who's in there? This time we asked the question about fungi. And this doesn't have anything to do with this talk. We've not been asking the question about fungi in this system enough. These are basically like lichens, these mats. There are so many fungi in there, and we don't know how they're relating. And shrimp are filter feeders, right? The fly larvae, they're eating everything. So we don't know, fungi are part of their diet, right? But we also don't know what other things are going on. We found a fungus that was a parasite for a nematode, and we were like, what the heck is that doing there? And then I'm talking to Michael Werner, and they find these nematodes, and then Mike and I start talking. Anyway, there's a whole world going on in these mats that we're only beginning to understand. So I'll back off of that for a minute and tell you that this guy was not in the mats in the lake years ago when we studied them, but it is now, and it is one of the most predominant photosynthesizers in the mats. So we initially thought it was Camptonema, which is a filamentous cyanobacteria. And we thought, oh, great, this is a pioneer species. It's like succession in the forest, right? It's somebody who starts the mats and then all their friends come to join the party. And maybe it is, but we now know that it's a eukaryotic alga. And I don't have the names on here, but I don't remember exactly what they are because this just came to us last week. But we're talking to some algal specialists, and they're like, oh, that looks like this or this. And so we look it up, and sure enough, so we're going to sequence the genome of this guy. So we know what it is, and we know what it does, and we can learn more about it. But isn't it pretty? Yeah. I have t-shirts made with this one. It is so pretty, but hard to isolate. You gotta pull those streams apart instead of just sampling a simple colony, so it's really challenging. Okay, so conclusions about the other study, that, the water column and the microbial mats are really different. And functionally, they probably can't stand in for each other. If we lose microbialite mats, I don't think the water column can help us with that, right? Most of the primary production is occurring in the mats, probably the secondary production as well. Microbialite mats, as you know, everybody in this room knows, are in the shallower margins so that they can get sunlight. And so they're more vulnerable to lake elevation decline. And was flipping ahead, the loss of these communities could reduce the net productivity of Great Salt Lake over an order of magnitude. I think that's really important to understand, and it gives us the argument that we need to keep water in this lake, because the whole ecosystem really depends on this primary production, right? Whether it's chemosynthetic or photosynthetic, that primary production is key to the base of the ecosystem. So I'll end by just saying thanks for funding. Both of these projects came together in a beautiful way, and I really appreciate it.
Thank you, Bonnie.
Yeah, sure.
I think in the name of time, maybe if we could ask Bonnie questions at the end of the meeting. I do want a filamentous algae shirt though,
All right,
when you
I'm
do
going
print
for it.
those. I would
Yeah.
love that. So we'll just go right ahead to Mike. Mike, if you're ready to share your screen.
There's some wrist shots, Mike.
This is a timely discussion following Bonnie's talk. Mike is looking at bathymetric LiDAR and how we can best utilize that tool to understand what's going on the bottom of the lake, including where these microbialite fields are. So whenever you're ready to take it away.
So the t-shirt idea came to mind as well. Great minds think alike. I just thought, wouldn't it be so pretty? Great conversation piece too. There's that over there.
I think you have to share your entire screen. Bonnie, it does scratch an itch to learn about photosynthesis again. I was one of those AP bio kids and I loved it. So I maybe am not the part of the US population that didn't understand it, but it's a fun part of my brain to go back to.
One of those things we think we know.
Yeah. Okay. Thank you to Marissa for giving me a few minutes today, squeezing me into the agenda. My name's Mike Vandenberg. I work here at the Utah Geological Survey. I am the head of the Energy and Minerals Program. And so the building you're standing in is kind of part of Energy and Minerals. So if you want to know about core and cuttings, we can do a tour afterwards and you can learn all about what's actually in this building. But we're happy to host meetings like this here also. So let's start off with, I am not a LiDAR expert. Let's just get that out there so everybody understands. We do have some LiDAR experts at the Utah Geological Survey. And so my colleagues, Emily and Kate and Kayla, Emily is a LiDAR expert. She's in our mapping program. Kate is in the groundwater program. She also has experience with LiDAR. And Kayla, who's in the back there, she works in the Energy Minerals Group and is a geophysics techie specialist. So the four of us are all working on this project together. And I also want to acknowledge Forest Fire State Lands and Great Salt Lake Advisory Council, who they provided some funding for the pilot project that we're going to talk about and some new. Okay. Bathymetric LiDAR, I don't want to get too far into the weeds, but basically, I think everybody knows about topographic LiDAR, which is planes flying overhead, shoot lasers at the ground, you get an image of the ground. Bathymetric LiDAR, you use a different kind of laser. So instead of the red laser for topo, you use a green laser. Green laser penetrates through the water column, reflects off the bottom of your body of water, whether it's a lake, a river, or the ocean, and returns up to the plane. So you actually get a image of what's under the water, whereas the red laser bounces off the top of the water. So pretty simple. Why do we need this? So I came at this from a place of trying to find some technology that would help answer some basic questions at Great Salt Lake. So I have a background in looking at the microbialites at Great Salt Lake with Bonnie. We've been looking at these over the years. And we were trying to better understand the distribution of the microbialites in the lake under the water. It's hard to see them when they get really deep under the water. It's hard to map. So we're looking for ways to better understand these distributions. So that got me sent down the rabbit hole of bathymetric LiDAR. This was back 2015, 2016. Bathymetric LiDAR back then was fairly new. It was new tech that was just coming out. It was mainly used in coastal areas. Nobody's ever used it in lakes. And I wrote my first tech team grant. It's submitted in 2017 to Forestry Fire State Lands, and they did not fund it. And I tried to get, let's see, we applied to NASA, we applied to this place, we applied to this. Nobody would fund a pilot project to see how well this might work out at Great Salt Lake. Well, then lake level went way down, and everybody got super interested in studying Great Salt Lake, so we took advantage of riding that wave of funding. So thank you,
Michael, I was just gonna say, I think your wrists have improved since then, so
I know,
it might be related. I don't know.
I've been doing my exercise. Good. Okay, so finally, it was, well, we'll get into the funding in a minute, but so my main question for trying to apply this to Great Salt Lake was related to microbialites, but it kind of evolved and we saw that this could answer several other questions. Emily Clever, who's a hazard geologist, mapping geologist, she wondered, can we see fault scarps in the bottom of the lake? So use bathymetric LiDAR to see fault scarps. But also, as lake level went down and we're looking at microbialites and we're standing on the shore and lake level is at a certain point and we compare it to older bathymetry, it's pretty clear that the older bathymetry in the nearshore environment is not so good. And this is, of course, where the microbialite population is. And so to relate lake level elevation to exposure of microbialites, you really need accurate bathymetry in the nearshore environment. We don't have that today. And so that was a whole nother reason to explore this technology. Okay, so the pilot study was funded a year ago. It was funded by the Great Salt Lake Advisory Council. UGS chipped in a little bit of money, so it cost $100,000, $75K from Great Salt Lake Advisory Council, 25 from UGS. We did 50 square miles on the west side of Antelope Island. So this black box there. We contracted with the company NV5 Geospatial. We collected this data about a year ago. So the data was collected in June of 2023. I won't get into the weeds of why we collected the data in June, but there is this period of time in the spring where the brine shrimp grades off the algae in the water column becomes much clearer than normal. And so we were trying to hit that clarity window because this data collection is really dependent on the turbidity of the water, the clarity of the water. So we were trying to hit as best clarity as we could get. We picked west side of Antelope Island because it had a range of environments that we wanted to test with the Bathy LiDAR. So it has microbialites, it has areas without microbialites, it has shallow water, it has deep water, you have the steep shelf. It has all these things that we wanted to see how well the bathymetric LiDAR would work in this environment. So the day we collected this data in June last year, the lake was at 4193.8. And so they collected the data and then it took them 3-4 months to process the data. So we didn't even get the data until October 2023. So let's talk about the results of the pilot. It's kind of a story of the good, the bad, and the ugly. So we're gonna start with the bad and the ugly. This is the area where we collected the data. And the really bad part of this, wherever there's yellow, we got 0 data because the water was too deep. So that means the laser couldn't penetrate all the way down to the bottom of the lake. It got absorbed somewhere in the water column.
What was the Secchi depth or the water clarity when you flew?
The turbidity was 8 point something, whatever the turbidity units are, which I can't remember.
Meters.
No, Nepal.
Oh, Nepal, yeah.
Nepal's
Secchi,
still around with 8 units.
Yeah, right. It's a mess.
Okay, so
Can I ask another question? Just while you're here at this point about laser penetration. My first entree into Great Salt Lake was looking at the biology's relationship to UV light. And I remember a lot of studies on saline lakes showing more UV light penetration. I don't know how lasers penetrate saltwater versus freshwater, but are there analogous bodies where this Bathy LiDAR has been used, or would the hypersalinity cause some problems with laser penetration?
So it's really about the clarity of the water, not the
Okay,
solute load of the water.
Yeah.
It's all about the clarity.
but the salinity
There
doesn't
is-
matter.
There is a correction because when the laser penetrates the water, it kind of refracts around.
Right.
And you have to correct for salinity.
I see,
Um,
but you can normalize that.
But yeah, it
Okay.
should work. Let's keep in mind, nobody has ever done this in a sailing lake.
Well, that's
So
what
we're also
that's what I
trying to
wanted to
learn
know,
as
yeah.
we go, and the reason we wanted to do a pilot study to understand some of those issues of what does high salinity do to the laser and to the data returns?
Yeah,
How is that going to compare to the north arm where it's saltier?
right. But with UV light, it definitely changes. So, I don't know anything about laser penetration, but with light penetration, it's really different than freshwater. It's the solutes.
She's.
Actually, breathing units.
Yeah. Yeah.
Okay,
That makes sense.
so overall, we got data only on 28% of this area that we shot. So we spent $100,000 and only got 28% of the data. Good learning experience. This here is the shelf rig. So we have shallow shelf on this side and then boom, it goes really deep. So it was not super surprising for me to see this, that we got no data in the deep water. Not super surprising, a little disappointing, but not super surprising.
Because that's the fault line.
Yes, this is the fault line. And this blue line was the water. So the level of the water the day that collected the data. So everything to the right of that blue water is topo LiDAR. So this LiDAR on dry land. Everything to the left is Bathy LiDAR. So LiDAR collected through the water column. So in general, in the northern area, we got depth penetration to about 4186, and in the south, we got depth penetration to about 4180. So at most, so remember water line, so to say 4194. So at most, we could penetrate about 14 feet of the water column, which is actually, it's not too bad. Okay, so that's bad. Now the ugly is the pink. And this was a huge surprise to me. The pink are actually void areas on the shallow shelf where we also did not get data for reasons yet to be determined. We don't really know. We don't know if there was something in the water column that day that they collected the data that kept the laser from penetrating down to the bottom. Maybe it was like foam on the surface. Maybe it was waves. Maybe it was a brine shrimp egg slick. I mean, we don't know. My hope is that it's something ephemeral. Like if we were to go out and collect that data again, that maybe we would get data in those void areas. We'll talk more about void areas. You can see this is a little snit. You can see these holes popping up in the data. Those are the voids. Okay, let's talk about the good, because there is some good. The data, where we actually got data, the data is absolutely amazing. I mean, the resolution of this data is absolutely amazing. This is just a little snip that's up here off of Bridger Bay, showing all sorts of random features. We got areas without microbialites, a big microbialite reef. We've got linear features that you can do profiles across that show some interesting topography. And the scale is really small here, but that's 0.2 meters. So that's like a foot. So we're seeing vertical resolution of less than a foot in this data, which is really, really good. Here, this ridge becomes an actual ridge. Here, it's more of a slope. But this ridge, again, is like 0.4 meters. So about a foot and a half. Carbonate spring mounds. So we've already made a huge discovery, maybe, I think. Way off of Ladyfinger Point, off Bridger Bay, all of these little domes showed up in our data. I was like, oh my gosh, what the heck is that? And I think those could be carbonate spring mounds. And so these are, again, some profiles. They're about 1 meter tall. They're about 10 meters in diameter. And these are bigger than microbialites. They're not microbialites, they're bigger than that. And we see features similar to this on the shores of Great Salt Lake that we attribute to Pleistocene water discharge. So we see tufa mounds, especially out by lakeside. So where the causeway crosses all the way across the lake. We've studied this tufa mound, we've published on it. It's about the same size as these things. And as far as I know, nobody's known that these are out here. And if these are spring mounds, this could be the first direct evidence of groundwater coming into the lake from under the lake. So we've all hypothesized that this is happening. We all think this is happening, but that is really hard to measure. It's really hard to find where it's happening. And this could be the first direct evidence that this is occurring.
Those are like the smaller version of what's out at Mono Lake. Those giant.
Yeah. These are going to be more like giant domes than tufa towers, with something similar to that. You can see from this aerial imagery here, when the lake got down to its lowest point in 2022, the tops of those things were actually exposed. And I really wish I knew they existed, because I would have taken a kayak out there to look at them at the time, but I didn't know they were there.
So Mike, there are some enormous ones on Gunnison Island that are maybe about the size of half this room, and they have layers. They don't look like the tufa at Lakeside. They look layered as if they had microbialite mats at one time, but they are enormous and they're collapsed domes. And every time we go out to Gunnison, I have million that they're really cool, but also not on Strom's Knob, but
hiking up from Lakeside towards Stromsdom are some of those big ones that I went to with Vicky and Jack. So I do, and they did also think they were Pleistocene. So
Yeah.
I think I
So
think
ma-
these guys are probably the same, huh?
Maybe these
Yeah.
are Pleistocene. That's
Cool.
really low elevation for a Pleistocene carbonate mound. We've
Yeah,
never
yeah.
seen them at that low elevation, but, intriguing nonetheless. I mean, again, these are 10, 15 meters in diameter, so bigger than this room.
Yeah.
They're
Cool.
A meter to a meter and a half tall. Okay. The other real positive, can we close this?
Oh, Jen, that's you're good.
Sorry, making the screen. Yeah. Okay, so the other positive and the other kind of utility of this bathymetric LiDAR, again, is to create more accurate bathymetry, especially in the near shore environment. So this is a comparison of the older bathymetry that was published by USGS in mid-2000s. That's the black lines here. So that bathymetry was generated, collected via sonar from boats moving transects. However, the boats can't get into the shallow water. And so a lot of the part in the shallow water is going to be interpolation. And so I think we're definitely in that interpolation zone when we get closer, like here, Bridger Bay. So orient everybody, Egg Island, Lady Finger Point, Bridger Bay. So that's the old data. So we can take the new bathymetric data that we collected and have the computer make contours with that bathymetric data. And that's what all the colored lines are. And you can already see some things jumping out, like this is a 4190 contour line, this tortuous red line through here, and the 4194 crosses it, the 4193 crosses it, the 4192 crosses it. So already we're seeing that the old bathymetry is probably not as accurate as data that we can get with this new bathymetric LiDAR. What I really like, and it shows the resolution of this data. So this mass of purple is the 4191 contour line. And this is a big microbialite reef out here that was exposed in 2022. And what this is doing is it's tracing around individual heads of microbialites. That's why that path is so short. And so you imagine, this is actually a picture in 2022 that I took of this area. And you have these individual domes, and guess what? They're all about a foot off the ground or a foot tall, right? And so the LiDAR data is drawing contours around individual domes. That's the resolution of the data that we're getting, which is amazing. So
That is so much higher resolution than the sonar stuff.
So if we can do this all across the lake, especially in the South Arm, that would be amazing. And if lake level goes back down, we'd have a much better representation of where microbialites are exposed, how big of an area of microbialites are exposed. Because right now, using the older contours, the data is just way off. Okay, so where we go from here? So last legislative session, so this is not the last one, but the one before, 2023 legislative session. We put in, Utah Geological Survey put in a building block to collect bathymetric LiDAR data across the lake. So we kind of did this at the same time we were doing the pilot study. You know, in an ideal world, we do the pilot study, we analyze the data of the pilot study, then we use that information to design something for the entire lake. Well, we wanted to take advantage of the year of the lake. Last year, 2023 was the year of the lake, and funding was flowing all over the place. And so we put in this request kind of at the same time. And it got approved, with some help from Casey Snyder and others. So it was an RFA. It is, the money is going to be coming out of the Forest Fire State Lines restricted account. I don't know if they're happy about that or. if they're still. Great. So we got 1.8 million in one-time funds from the Forestry Fire State Lands restricted account. Summer of 2023, we signed, UGS signed an MOU with Forestry Fire State Lands to kind of administer this project. October 2023, that's when we got the pilot data back. Spent a couple months analyzing that data. I prepared a presentation for the American Geophysical Union Conference that was in December of 2023. That's what these photos are. And that poster is on the back, on that board back there. So you can look at that after the meeting and if you want to talk more. So then this winter, we started the procurement process to collect more data. And the way that we had to do this, let's see, so LiDAR companies are already under state contract with the state of Utah, several of them. Several of those LiDAR companies have the capability of collecting that LiDAR. And so it makes the process for us a little bit easier as these companies worked on state contract already. So I sent out what's called a solicitation of qualifications to 3 companies. I did that in March. I gave them a deadline of April 26th to submit proposals back and a statement of the qualifications back to me. We got those back, and like during that process, I had meetings with all 3 companies, just kind of explaining what we want. We got the proposals. Now we're having second meetings because, of course, the proposals, we just have a few questions. And so we had our first one yesterday. We have 2 more on Friday. Then we will need to decide on which company we want to use to collect more data. Let's talk about the data collection. This is a super, super challenging project. And this is what we tried to convey to these companies. This is not going to be straightforward. This is going to be complicated. We're kind of charting new territory in this data collection. And then, of course, we don't want to just waste money. We want to make good use of the money that we have to collect data that is going to be useful. So when I put out the solicitation to these companies, I made some boundaries around areas that we would like to collect this data. And that's what these maps show here. So the all-in, well, first, I separated Great Salt Lake into 4 areas: South Arm, North Arm, Farmington Bay, and Bear River Bay. Okay, so South Arm and North Arm, we have boundary that goes all the way out to the high water line. That would be kind of the holy grail if we had unlimited money. It'd be super cool to collect topo LiDAR with the battery LiDAR from high water line. And then using the data from the pilot, we cut it off at 4180. So that's why there's the giant hole in the middle. We don't want to waste money collecting data in the deepest part of the lake when we know it's probably not going to work. And the USGS, the bathymetry in the deeper part of the lake is actually not that bad. It's the nearshore environment where the data needs improvement. It's not the deep part of the lake. So that's why we carved out the center. If that is going to be too expensive, we have another alternative in basically collecting data in this shaded zone. So it kind of creates a halo all around the lake in the nearshore environment from 4194 to about 4180. And the reason for that is we've already collected topo LiDAR data in 2016 all the way out to the high water line, all the way around the lake. So if we were to do that, we'd be collecting new topo LiDAR data, which maybe we don't need. I think it would be interesting be some application for it. But this project, we probably just want to focus more on battery LiDAR and less on LiDAR. So that's kind of the cheaper alternative that we're leaning towards. Then we have Bear River Bay, Farmington Bay, and these were areas that other people have more interest probably than I do. But I've been working with like Ducks Unlimited. It's really interested in collecting data in wetlands areas. So these areas got added through the Ducks Unlimited, Casey Snyder connection to help fund this. And so the wetlands areas also have a lot of challenges. You know, so we have lots of bare ground, so areas where you get topo, lots of ponded areas, lots of vegetation. We really don't know how well the battery LiDAR is going to work in any of these areas. One other thing I learned is if your water is too shallow, the laser, the red laser bouncing off the surface of the water looks very similar to the green laser bouncing off the bottom, lake bottom, and you can't tell that difference. You need like some kind of bigger delta to be able to tell the difference between what's bouncing off where. And so if you get too shallow, it also doesn't work very well. And so in these ponded areas of the wetlands where it's all fairly shallow, it might not work very well. So we're still trying to figure out the best way to deal with the wetlands areas. These are huge areas. You know, maybe we do some kind of pilot scale type test flights in the wetlands areas, see how well it works before we commit to doing a huge area. Then we have Bear Lake. So Bear Lake got tacked on to all of this. And my interest is at Great Salt Lake, but Bear Lake got added to this. Bear Lake is much more straightforward. The water is very clear. And so adding Bear Lake to this does not create a huge expense. We do have some jurisdictional issues of, do we spend Utah money on collecting data in Idaho? But DNR, I guess, is talking about this and working through those issues. So this is where we're at in this process. I would absolutely love any feedback from anybody here on kind of the priorities, you know, how best to tackle this. Again, the point is we want to collect useful data without wasting a bunch of money. So we're, like I said, we're trying to set priorities because we don't have enough money probably to do even the smaller areas of the North and South Arm, the two bays and Bear Lake. We're going to have to pick and choose or find more money. Yeah, questions?
Why was Bear Lake thrown into the mix?
It was thrown in when we did the building block because of this issue with geologic hazards and faults. So there's big faults on the east side of Bear Lake. That's why I got attacked out. So, I mean, maybe Forestry Department of State Lands and UGS can get together to see what the priorities are at Bear Lake. USGS has done bathymetry at Bear Lake, how similar to how they've done it in Great Salt Lake. And because the lake is much deeper, you get the boat closer to the shore. The bathymetry currently in Bear Lake is not that bad.
And correct me if I'm wrong, but I believe that the way this was gonna work is whatever funding was left would go to Bear Lake, right? It's not. Okay, I don't believe this was going to be prioritized.
Mike, I think just because we still have Tim left on the agenda, I know you wanted to have more of like
That's right.
a discussion, and maybe we could have an auxiliary tech team meeting for those that are interested in talking about this with Mike. But I would definitely encourage you to engage with UGS, talk to him, look at his cool poster.
If anybody has any thoughts, suggestions, please reach out to me. I will be here after this meeting. We can talk looking at the poster and I can show you more things on the poster, but I'm very interested in other people's perspective on this because I don't want to be the sole decision maker spending $1.8 million of Ben's money.
Yeah, I wish.
Good job.
Thank you, Mike.
One last question, could the data gaps be attributed to groundwater flowing up the fall and the data correction is misleading you because of the density difference?
What we see when we look at this like point scattered cloud of data is there's no bottom return. And so the laser is getting absorbed somewhere in the water where it is.
Thanks again, Mike, and sorry to, cut off
No, no,
question time. And sorry, Tim, to, leave you with such a small.
I'll be fast. So, Marissa, I asked Marissa to give me just a few minutes to talk about implementation of the Great Salt Lake Strategic Plan and what the Great Salt Lake Commissioner's Office is focusing on. I'm going to go through it really fast, give you guys a chance to ask a couple questions. A large part of what Commissioner Steed and I have been doing over the last 6 months is education. It was both the development of the plan, but then getting out. I think we're giving like 5 to 10 talks a week.
Welcome to my life.
Yeah, I know. So everything you guys have been doing, the Great Salt Lake Issues Forum is great. We're trying to add on to that and really try to elevate the conversation and just build more support across the state, the Great Salt Lake Basin, but then and in DC. So both Brad and I will be in DC next week having similar conversations with our federal partners. We are also working on a 30-year in the strategic plan. We committed to developing a 30-year more detailed plan for how do we get the lake to a healthy range and sustain it there. That we are actively working on that with the Great Salt Lake Strike Team. Will be Laura and Water Resources have graciously put into 2 studies that they are doing with the Great Salt Lake Basin Integrated Plan. We'll be pulling data out of that in November. There's really 3 main areas that we're looking at. We're identifying what's the baseline increase in water flows to the lake or decreases in direct depletions from the lake that we need to get every year over the next 30 years. We're trying to pick a realistic number that we can get to in the next probably 5-6 years, and then see how the lake responds. We are also we'll be working closely with Forestry, Fire, and State Lands, the Salinity Advisory Council, on adaptive berm management and design. That'll be a critical piece. We learned a lot from last year when the berm was closed, and we learned a lot this year when the berm was not closed. So we need to really lean into berm management. We're part of that. Active berm management will be part of that. And then the last 2 years have also given us a really good example of how do we take advantage of wet water years? How do we get as much water to the lake as possible? During those years, we're already in conversations with the different water districts, Pacific Corps, to learn after this runoff season what worked, what didn't work, how much extra water got to the lake. This is really the first year that we've been measuring. We've been asking them to measure those flows. And then working with water rights, they've been working with Utah State University on the gaps analysis, because there's clearly huge gaps in our data about, very selfishly, just how much water is getting to the lake. We've got a lot of gauges that are far from the lake, relatively far from the lake. So how do we get better data? And then Water Resources took the lead on also applying for a Water Smart grant with the Commissioner's Office for airborne snow observation in the Weber Basin. So how do we get better data on how much snow we have going forward? So that's a big lift right now. We're also shifting towards figuring out how to lease water. Split season leases. I'll be meeting with a lot of conservation districts, had a great meeting with the Great Salt Lake Watershed Enhancement Trust, working with Wildlife Resources staff as well on we need to start utilizing all these new statutes about saved water that Water Rights is taking the lead on. The funding, we've had $61 million appropriated to either the Commissioner's Office or through Forestry, Fire, State Lands to the trust. We need to start getting water leased and secured. And then once we've identified that baseline amount, that's what we'll be building towards with the trust and securing both permanent, but also seasonal leasing and split season leases. And then working closely with water rights to make sure that those go through the change process. Investing in science. We have a dust forum this week, bringing together water, land, and air managers with dust scientists from universities as well as agencies to really pull those two together, identify what we don't know, how to fill those gaps. Groundwater, we've invested in addition to the tech team and UGS and understanding more about groundwater inflows into the lake. That's clearly a gap. And then investing, working with the Division of Wildlife Resources on Wilson's Phalarope, snowy plover, with the petition to list the Wilson's Phalarope with U.S. Fish and Wildlife Service. We're trying to get our help invest there to get a better understanding of their life cycle. And obviously, what we've been talking about today will play a huge role in that. And then the last two are, one of the key roles in my job as deputy commissioner, but also the commissioner's office, better coordination of everything that people are doing on the lake. We'll continue to work on that and improve that. We will be bringing on a couple staff in the new fiscal year to help with that, as well as to go sit down on kitchen tables of farmers and others to say, how do we help all of us work together to get water to the lake? And then securing long-term funding. That's another part of our conversation this in DC, but we committed to coming up with a long-term budget for what it's going to take to get the lake to that healthy range to sustain it. It's not going to be cheap. We've clearly we have not appropriated enough. We wanted to telegraph that in the plan to legislative leaders that we will be coming back with this probably in year 2, and year 1 of the commissioner's office being stood up is July, so it'll probably be something like that next fiscal year. So that's where we're at. Any questions in how did I do on time?
Oh, excellent.
Okay.
Yeah.
Whoa.
You can tell you give a lot of presentations because you've really nailed it down.
Yeah. Are you communicating all of this with the other, with Idaho and with Wyoming and in some sort of a ongoing conversation through the commission?
Not really yet.
Yeah,
So we will probably be working closely with water rights on that, just with the Bear River Compact Commission. And, but we've talked about it, but we just haven't gotten around to it.
Yeah.
Because we would love to see some type of federal program that would fund additional conservation within the Bear River system writ large.
Right.
That's consistent with the provisions of the Bear River Compact settlement. So, but yeah, we haven't. It's a great question, Lynn. We're not there yet.
Yeah.
So, yeah.
Doing a lot.
Yeah.
Thank you.
Any other questions? And if you have any suggestions or dad jokes, please just you can email me, text me, any of those things. So.
Thank you, Tim.
Yeah, thank you.
Yeah, Tim, we're forcing Tim to come to all these meetings, so, we can continue to discuss the strategic plan as it relates to what we do as a tech team body. So whether he likes it or not, he'll be here at our next meeting as well. So if you have questions then, great.
So if I can add one more thing, when we have the draft 30-year plan ready, at this point, I'm planning to take that to the Great Salt Lake Advisory Council and so that to get public comment, get feedback. We didn't have time to do that on the strategic plan, given that I started in like 3 weeks before the plan was due to the governor and the speaker and the president. And so, but we are planning to get that out for public comment once it's ready. And we'll be working closely with Forestry, Fire and State Lands, water rights, water resources, water quality, the agencies as we draft it and make sure that it's teed up and it's in a place where we're all comfortable with where we're trying to go. But it's really designed to, here's how we get the lake in that time period. Here are the key levers that we need to pull to get the lake to a healthy range, and get more specific about decreasing depletions from the lake and getting more water to the lake. But yeah, so you'll have a chance. I don't think I'll bring it back to every single one of the Laura's advisory council or steering committee to the tech team. I think we'll use the Great Salt Lake Advisory Council as that public forum, but look for that opportunity. Probably late summer is when I think we'll have a framework of that will then plug in data from Laura's studies and others after that.
Great. Thanks so much, Tim, and thanks to all of our presenters for squeezing your definitely longer talks into a really short time window. So we have about a minute left. I just kind of wanted to open up the floor. Does anyone in the room have any updates, issues, questions, comments, concerns, anything you'd like to share with the group, other meetings that are happening here soon? Anything? Yes, Wayne.
I think I mentioned this before, but there's an International Society for Salt Lake Research who's sponsoring a conference in Turkey in early November. They recently extended the early registration deadline till close to the end of this month, I think. So if you're interested in that, you can talk with me or look on their website and find the information. It's in a very nice place, pulls together people from all over the world and are working on Salt Lake. So it's really nice venue.
Plus the cool kids are gonna be there.
Thanks, Wayne. Sounds like a really cool trip and conference. Lynn, 2026 dates?
Yeah, 2026 dates. What is the Wednesday following Thursday, May the 7th?
Almost got it.
Yeah, that'll be the dates of the 2026.
Awesome. Thanks, Lynn. Anyone else? Anyone online? Speak now. All right, well, our next tech team meeting is on September 10th. It is also in this room. Thank you, UGS, for letting us be here. And thanks to all of you for walking and or driving out here. Hopefully it's a nice break in your day. And more annoying emails to come from me on our agenda and such. And as always, feel free to reach out to me if you have any tech team related questions or you want, squeeze in on an agenda. Definitely just talk to me. So thank you all for being here. And what is it? We go. Yeah. A link up. Thanks, guys.
Wait, Brian and I, at least I'm going to plan for that one.